Thank you very much for the kind introduction, and thanks to organizers for the invitation, like David said, to talk about some of my work and what I'm excited to do with this beautiful next-gen data. I actually have a couple thank you slides. The first is mostly UC Berkeley, UCSF people. I'm going to talk about collaboration that started maybe around 8 months ago and kind of some of the results of this project. And the second thank you is what I call our tractography team at Vanderbilt. These are students mostly in Bennett Lemon's Massey Lab who have done a tremendous amount of processing that you'll see today. So I have a 3-minute primer on diffusion MRI. I know most of you are hardware and software teams, so we're going to go through the basics. Proposing to study microstructure and connectivity using just the diffusion of water molecules in tissue. And I think that by studying this, we can perform a virtual in vivo histology as if we're sticking tissue directly under a light or electron microscope to measure features of size or density. And second, a virtual in vivo brain dissection, again, entirely non-invasively, as if we had a cadaveric brain in front of us that we could cut up and study these fiber pathways. And to explain why this gives us these, I like to show cartoons of the different cities we're in. This is campus, and these dots are people. These might be us watching this talk right now. There may be students walking around campus, and these yellow dots are maybe people leaving a bar or their favorite restaurant.
Demonstrate.
So if we're magically able to follow these people over time or follow the random walk or random drunken walk, we could learn a lot about the city or campus, maybe the size of this building, maybe the speed of these students, or different boundaries across campus where they can go or where they can't go. And most interesting, on University Avenue, we could start to, start to guess the directionality of these streets. And if we follow people long enough, we could start to make connections all across campus in the city. So obviously this is
where the good restaurants are.
I'm sorry, I've never been here.
We have I wrote up something to hand out to people for recommendations.
So I have another map later. Maybe that's a better place to visit. But so obviously just a giant metaphor for tissue microstructure, measuring the direction and magnitude of diffusion. We could all of a sudden measure things like cell size, cell density, and in the white matter, we could start to guess its orientation. So instead of ink in a beaker, we acquired diffusion-weighted magnetic resonance images that are just images sensitive to diffusion in a bunch of different directions. And then we're going to use this to perform our histology in our dissection. Our dissection starts with estimating that direction of greatest diffusion in every voxel, creating 3D vector maps that we can step along in all sorts of smart ways, basically playing connect the dots. And these generate streamlines that represent connections from one area to another area in the brain. We did this throughout the whole brain, generating the human connectome. And then one thing that I've done and my lab does is dissecting or pulling out or segmenting specific fiber bundles or pathways, maybe projection pathways, commissural association pathways that are all functionally interesting for some way, maybe related to cognition, senses, reading, language abilities. And once we've segmented these, we can study these. We can extract certain features or potential biomarkers, maybe macrostructural features of the size, maybe measuring atrophy, lengths, surface areas, diameters. And second, microstructural features. For this talk, I'm really going to focus on, many of you have heard these features from DTI, fractional anisotropy. That's a measure of coherence of tissue, where you may have a high FA or a high coherence, or it may lose its coherence and we may have a lower FA. And second is measures of diffusivity. This happens to be radial diffusivity, orthogonal to the direction of fiber bundles. And you can imagine if we have a low exonal density or low myelination, there's more room for water to move, and we may have a higher radial diffusivity. So the whole point of this is to show you that this is a multi-step process where we can extract functionally relevant white matter pathways and pull out biomarkers that may be useful or interesting. And that sets the stage for the rest of the talk. I'm going to spend a few minutes talking about my work, not next-gen data, but very typical, mostly 1.5 or 3T data, understanding changes across the lifespan. Then I'll talk about what I'd love to do with this next-gen data and what I'm excited to do with these beautiful data sets over hopefully the next few years. And then we're going to take a few steps back and address some of the challenges that we've actually seen in earlier talks. So white matter across lifespan, this is pretty intuitive. Most of us know the brain changes from infancy, development, young adulthood, and then aging. And over the last couple of years, one of my goals has been to describe what normal changes are so that hopefully we can identify atypical or abnormal changes and hopefully identify them earlier. And we were motivated by this idea of pediatric growth charts, where you can take any individual at a certain age, and you actually place them in the context of the rest of the typically developing population. So you may say someone's 2 years old, they may sit at the 60th centile, or oh no, they sit at the second centile, something is abnormal and worth looking into. So we would like to do this with white matter brain charts. This is work by Michael Kim, a PhD student, who's actually done a tremendous amount of data processing. He's taken about 50 different large cohorts or consortiums spanning 0 to 100 years of age, over 70,000 individual data sets. I think it's something like 8 million individual images. And from every scan, he's actually pulled out about 70 white matter pathways and those microstructure and macrostructural features from those pathways. And what he's going to do with these 70,000 data sets is fit all this data to a gamless model, a generalized additive model for location, scale, and shape that's exactly like those pediatric charts, where we can now take a new brain and put them on this chart where we can now say this white matter pathway is normal or abnormal, or sits at the 60th centile or the second centile. And we've identified 3 areas where we're starting to use these brain charts, and I have one slide on each. Defining normative trajectories, identifying developmental milestones, and detecting deviation from normal. So first, just defining trajectories, we can take different pathways. These just happen to be really common projection, association, and commissional pathways. Plot all the data points. There's tens of thousands of data points in here, and actually create these charts that describe the normative trajectory. They're sex stratified, so we have the trajectories for male, females. I think we're showing the 25th, 50th, and 75th centiles, but you can already appreciate differences across the lifespan and differences across the pathways. We can start to understand where variation occurs across the population, where our brains may be more or less similar across the lifespan. And we could start to characterize typical rates of change, maybe growth during infancy and atrophy during aging. The second application is just identifying developmental milestones. I like to show this picture. It's just 2 different pathways, and we just plot the lifespan trajectories of all of these features. And you can appreciate that these have different rates of change during development, different atrophy during aging. And even within the pathway, not all these features are changing at the same time. So there's different microstructure and macrostructural processes that happen at different times and differently in different pathways. And we show these relevant diagnostic age ranges for a few diseases and disorders, just to emphasize that our charts go from 0 to 100 years of age. So they're potentially relevant to identify abnormalities in whatever application you're interested in. And then this right chart here just plots some of our features against a very well-known waves of myelination that occur throughout the lifespan. And we can show that that radial diffusivity measure that broadly reflects myelination also has waves or epochs or changes across lifespan. And we can maybe start to rank across all the pathways when these waves occur and when typical waves occur and when typical atrophy starts to happen. And all this leads to the last application that we're really starting to kind of dig our really early investigations into, is to identify deviation or atypicalities. We can take healthy controls that by definition are a uniform distribution on these centiles. We could maybe look at individuals with mild cognitive impairment or with Alzheimer's disease, and we see that on average, they're at the extremes of our centiles with lower coherence, lower myelination, and lower volumes and areas. And this plot is actually just one index across all 70 pathways and all 20 micro and macrostructural features. And we're really starting to ask how we can use these to identify outliers or patterns of outliers. And most importantly, identify patterns in individuals that are cognitively normal, but we know convert to some sort of cognitive impairment. So I don't have any other work here other than showing the single density plot that shows that there are at least outliers as you progress along this cognitive impairment progression. So that's my work. Identifying normative trajectories, identifying atypicalities. We'd love to actually use this to do brain behavior mapping. But what leads into this beautiful high resolution 7T data is these hard to track pathways that are functionally interesting that we just cannot track with normal 3T or 1.5T data with regular gradients. So there's a lot of opportunities with diffusion imaging data. This was the map we started with that apparently does not include the best restaurants in the city. As we saw, Joseph talked about earlier, this higher SNR affords higher spatial resolution and higher B values. That all of a sudden changes the picture. This is actually just zoomed in, so we're not moving across different streets here. But the picture's completely changed. Instead of buildings, we can look at rooms. Instead of the entire campus, we can look at compartments across the campus. And instead of major streets, we could look at side streets and even sidewalks of the brain. And I've talked to David, Joseph, and the team here, and I try to describe that the data is so beautiful, you don't even need to do tractography to appreciate where these pathways are. So I have this history of tractography, generally kind of invented around 1998, 1999. But we're going to go 10 years before that with the idea of MR neurography, where you just look at projections of these diffusion weighted images and different pathways all of a sudden stand out. We can immediately do that with this data. This is actually extremely minimally processed. And just taking a projection of this diffusion weighted image, we can immediately see where the arcuate fasciculus is, where this beautiful claustrum is, the cortical spinal tract, even different parcellations of subcortical nuclei. Again, we're not even using tractography, and I can tell you where these pathways are. What I'm most excited about, I hope you can appreciate it, are, we saw this with Joseph earlier, these superficial white matter, short association fibers that actually make up 80% of the connections in the brain, but 99% of our literature are these long-range pathways I talked about earlier. We can go one step further, showing these red, green, blue color maps. In a typical image that I'm used to looking at, well, first they point in the direction of diffusion. Red is right, left, green is anterior, posterior, blue is superior, inferior. These already look pretty. But when we look at this with the NexGen7T, you can immediately appreciate these superficial white matter fibers, the beautiful striations of the optic radiations, again, the arcuate fasciculus. And I'm not an anatomist at all, but you can train someone like me to immediately segment all these beautiful brainstem pathways that are just so small you can appreciate these at 2 or 2.5 millimeter resolution. Where all of a sudden we have these projection sensory and cerebellar pathways that you can pull out and segment and study. Just some more benefits, some more still qualitative images. Like I said, a virtual histology. The left is a brainstem atlas from 100 years ago. Maybe not 100 years ago, but you could see these really small strands of pathways that essentially go orthogonal to the main cortical spinal tract. And this is what Joseph was talking about, were these multiple fiber populations in a voxel that you just can't see at low B values. And all of a sudden with these data sets, you can appreciate these left-to-right fibers through this really big pathway that dominates diffusion direction. So even without tractography, again, we could see this histological, these images that represent these histological atlases. And again, you could do connect the dots in your mind and see these superficial white matter pathways. So what do I want to do with the data? I want to study the long-range white matter pathways. I want to study the superficial white matter, these short association fibers. This is work from my postdoc, Matt Amendola, who's come up with protocols to segment the prefrontal cortex, different connections of the prefrontal cortex. But we're slowly realizing we need high resolution data to do this. And it's just not possible with data that we can acquire with our scanner and our gradient system. So we're very excited to start these processes on the NexGen7T data. And the brainstem pathways, these small intricate fiber systems that I did make a tractography image. I couldn't insert it in time. But like I said, these are so high resolution, you can just draw where these pathways are. So the final part, the challenges. I'd like to describe the tractography process as a multi-step process where every step in the process presents a new challenge. Uh, some of what we've seen, motions, artifacts, distortion, the number of the B value, the number of directions you acquire, how you generate these streamlines, how you segment these streamlines, how you quantify connectivity. All of these ultimately impact what we really want to do, which is make inferences across the population or individual subjects. So we're going to take 3 steps back and look at just one artifact, distortions. As we've seen, distortions are a problem because of the high field, and we're pushing the resolution with these EPI sequences. We know these affect the geometry. We know these affect the signal. They lead to incorrect orientations and make tractography hard. The good news, we've heard about the hardware and the software image reconstruction. There's also post-processing techniques to solve this. You can acquire your diffusion data. You can acquire a single reverse phase encode image that's essentially equal and opposite distortions. Where an image was pushed, it's now pulled, and estimate the middle ground, correcting distortions. You can do this in the left to right directions, pushing the image left and right and estimating the middle ground. You can actually collect half your data set with one reverse phase encode, the other half with the other. There's reasons that this better helps you mitigate eddy currents in addition to distortion. And finally, you can do 4-way phase encoding that also has additional artifact mitigation like ghosting. So these have all been developed, optimized, validated at 1.5 and 3T. We just ask whether you can do this and whether they work at 7T. So there's a lot going on. I'm going to go through this in 1 minute. We designed an acquisition that's extremely oversampled and lets us create all these subsets of protocols that are really common at 3T. A single reverse phase encode, 2 sets of reverse phase encode data, or that 4-way acquisition. And then we look at every possible combination. We look at geometric accuracy, we look at reproducibility, we measure ghosting, and the overall SNR of these images. And I'll show the qualitative results. Obviously, if you don't correct your data, it does not align with, for example, structural boundaries at T1. With a single common reverse phase encode, we get better correction, but it's still not perfect. And the more reverse phase encode data you acquire, the better you can correct these images. And this correction lets us do the tractography that I want to do. For example, we cannot do that superficial white matter tractography without it, but with 4-way, all of a sudden we can. So we're kind of coming up with recommendations and how we'd like to approach this in the future to do those tractography of the brainstem and superficial white matter. So that's all I have. I'm very interested in studying the lifespan, very interested in working with this data for these hard to track fibers, but we really do need to understand every step of the process and really optimize the tractography process in addition to the image acquisition. Thank you.